Well, welcome everybody, and thank you very much for joining us this evening. My name's David Adams. I'm head of the College of Medical and Dental Sciences at the Medical School, and it's my pleasure as head of the college to host the Bladder Cancer Research Centre. I want to give a particular thanks to all of the donors on the call who have helped us turn this into a reality. We're really grateful to you, and I think you'll see over the course of the next hour or so that your donations have been put to really good use. And I'm delighted that one of our donors actually will be speaking shortly and sharing with us why he decided to donate to the centre. So a year after the launch of the centre, you'll hear from key researchers and on the progress that's been made. I think that despite all of the problems that we've had over the last 12 months, the progress has been remarkable, and the centre is proving to be a terrific success. It's been a difficult period to continue with any research, including cancer research, but we are beginning to come out of the pandemic. There are still problems, but research is active again across the university and across the college. And I'm delighted to see the Bladder Cancer Centre really get up to full speed once again. I think this has been a real success and will be an even greater success in the future. And for me, there's been a particular interest in bladder cancer. When I first qualified as a doctor, my surgical house job, I looked after a wonderful patient who just happened to be the landlord of the local pub, which was helpful as well, but with bladder cancer. And he came in repeatedly for repeated treatments. And I just saw over a period of 6 months when I looked after him what a devastating disease this is. And of course, many of you will be aware that this is very much a Cinderella disease. It doesn't really get the attention it deserves. It certainly doesn't get the research income it deserves. But any of you who have been touched by bladder cancer will realise what an awful disease it is. But it's a disease that Rick and the team here are making real progress towards diagnosing early, towards developing interventions and new treatments. And so we hope that in the future, this disease will be far less of a problem than it currently is at the moment. So I'm really thrilled at the way the centre has developed. I think you'll enjoy hearing from the team this evening. Once again, thank you all for joining us, and I'll hand over for the rest of the programme. Thank you.
David, thank you very much for that introduction, and thank you very much for your support in enabling us to establish the Bladder Cancer Research Centre at the University of Birmingham, and your ongoing support and encouragement since we launched just over a year ago now in November 2020. Before we kick off, I'd just like to remind you a little bit of Zoom etiquette. This event is being recorded, so I'm sure you've all been asked to press OK to say that you're happy for it to be recorded. Only the speakers will be captured on the recording, and anyone who decides to ask a question live in the question and answer session at the end. If you don't want to put a hand up and ask a question live, then please feel free to submit your questions via the chat box, and we'll do our best to get through as many of the questions as we can. And if you do want to pose a question live, then be ready to unmute rather than hearing a chorus of "you're on mute." So great. So I think it's really important to again reiterate what David has said. Thank you to all of the donors, many of whom are on this Zoom this evening, all of the donors who've made the research centre possible. We've had a really busy year, and to a certain extent that's why we didn't actually have this event in November when it was actually 1 year. We've just been so incredibly busy that we've had to leave it a little bit later. Some of the exciting things you'll hear about: we are working with a commercial partner to bring a diagnostic urine test for bladder cancer to the market. Also this year, we've been awarded £1 million in research funding from 3 different funders for 3 different projects that we're very excited about. Finally, but certainly last but certainly not least, I'd like to say a really big thank you to all of my team in the research centre who have worked incredibly hard since we launched. Everyone has been personally affected by COVID one way or another, whether that's prolonged periods of homeschooling or illness themselves, etc. It has had an impact on our work, and we've remained very, very productive throughout. So thank you very much to everybody. I know we're hearing from the theme leaders shortly, but obviously there's a whole team behind them as well that I would like to thank. But before we do hear from the team, we're going to hear from a great friend of the university, a great donor, Michael Chowan, who has his own bladder cancer story to tell. Michael, can I hand over to you, please?
Thank you. You may be surprised to hear that someone who has experienced bladder cancer twice considers themselves lucky, but I am, and for 2 reasons. Firstly, I consider myself lucky because in May I will celebrate 15 years since my second and very expensive cancer operations. Only around half of people survive bladder cancer for 10 years, but 15 years later, here I am, I survived. My first bladder cancer diagnosis was 18 years ago this month. Initially diagnosed as an infection, it was correctly re-diagnosed, and a week later, in Brighton, it was removed. This was followed by a painful period of immunotherapy, and then only the second portable PET scan ever in Sussex confirmed my good fortune, that I was clear. My radiologist asked if I might wish to share my good fortune, and thus started my involvement in cancer research and imaging. My second bladder cancer diagnosis was a warning from a panel of Sussex medical consultants that I was at risk, only at risk, of a second bladder cancer diagnosis. Despite no symptoms, I approached the specialist, Professor Richard Hauptmann in Ulm, Germany. He examined me and bluntly advised me, "Michael, you have bladder cancer. You are dying. I have booked you in for major surgery 1 week from now." 8 days later, I spent a day in theatre, a week in intensive care, and 6 weeks recuperate. I returned just in time for the official opening of the Clinical Imaging Science Centre in Sussex, with one of the first PET scanners to be installed in the UK. More good fortune to share. And secondly, I know I was lucky to be able to get access to the most cutting-edge treatment at the time. I could afford private healthcare, which meant a lot for an underfunded condition like bladder cancer. I want to make sure that everyone facing the same diagnosis gets the funding, research, treatment they deserve. Right now, there are around 40,000 patients in the United Kingdom living with bladder cancer. That's more people than would fill my local football stadium in Brighton. This 40,000, though, doesn't include the families, their friends, nor their dependents. So my wife and I were thrilled to make our first donation to the Birmingham Bladder Cancer Research Centre. The centre's work will mean that the right patients are matched with the right treatment at the right time. New drugs will be developed, and symptoms will be diagnosed faster. The centre is on the brink of making groundbreaking discoveries. These will change and save the lives of so many people with bladder cancer. It will also protect their families and their friends, and other dependents, and others dependent upon them from the heartbreaks of bladder cancer death. And just thank you all for allowing me the privilege of being able to share those thoughts with you. Thank you.
Michael, thank you very, very much for, sharing a story, a very personal story. Thank you for your support of the centre, and I hope you enjoy listening to the team.
Well,
you and I have chatted for a long time, and now it's your chance to hear the rest of the team. So thank you very much. And I'd like to now head to Dr. Doug Ward, who leads on proteomics and biomarkers. The floor is yours, Doug.
Thank you, Rick. Okay, can you all see my slides? Great. So I'm going to talk very briefly about Inform Bladder, which is a urine test we've been developing to detect bladder cancer. So flexible cystoscopy is the way that bladder cancer is usually detected and has been detected for decades, but it's very unpleasant for the patients and it's very expensive for the NHS to perform this technique. So we thought, well, could we develop a urine test which could at least partially reduce the number of cystoscopies that need to be performed? And we started out based on three principles, really. One is that bladder cancer, as with all cancers, is a disease of the genome. So there are alterations in the DNA sequence in bladder cancer cells. Modern sequencing technologies allow us to sequence millions of DNA molecules in parallel, which is a very efficient way of detecting these mutations. And bladder cancers shed both DNA and cancer cells into the urine. So the first thing we had to do was select which mutations would be the best to base the test on. We spent a long time sequencing nearly 1,000 tumors until we arrived at a panel of 400 or so mutations across 23 bladder cancer genes. And our data showed that this should be sufficient to detect, as near as makes no difference, 100% of bladder cancers. So the next step was to evaluate the test using urine DNA. And to do this, we teamed up with a local company called Nonicus, who have a very clever way of taking the DNA and preparing it, making a sequencing library that we can then capture the 23 genes from, and we can sequence the genes. And we're looking for single letter changes in the DNA code. And then the software that we use to analyze the data tells us whether there are mutations or not in the sample. If there are no mutations, it's a negative test result. and it's very unlikely that the patient has bladder cancer. If there are one or more mutations in the urine DNA, it's a positive test result, and it's highly likely that the patient does have bladder cancer, or at least that's the theory. So over the last year or so, we've set about testing that theory, and we've looked for mutations in the urine of 165 cancer patients, and we detected mutations in 144 cases, so that's 87% of cases. And we looked for the mutations in the urine of 264 patients without bladder cancer, and we correctly identified 223 of those. So we have 85% of the non-cancers correctly detected. So these performances are actually very, very similar to the performance of the gold standard technique, flexible cystoscopy. How might this be useful to clinicians and patients? Well, each year in the UK, about 100,000 patients are investigated for bladder cancer, primarily because they have blood in their urine, and they will have 3 tests: flexible cystoscopy, imaging, and urine cytology. So that's 100,000 cystoscopies to detect 10,000 cancers. So that you could argue that's 90,000 unnecessary cystoscopies. So we have proposed that if we replace flexible cystoscopy with our test, Informed Bladder, and we do the biomarker test, the imaging, and the urine cytology, patients can be discharged if all 3 tests are negative. If any of the tests are positive, they go on and receive a cystoscopy. And doing this triaging up front of cystoscopy could save up to 50,000 unnecessary cystoscopies each year. So this study, which is now funded, is a feasibility study. It's not interventional. We just want to check that this is feasible and that it's safe and that everybody benefits and that it doesn't result in any patients being missed. The other context, as Michael mentioned, is that patients who've been treated have to have surveillance. So we've also said, how does the test perform in the surveillance setting? What we find is we can still detect cancers well. We detect 86% of recurrent cancers. But unfortunately, we get a lot of positive results in patients who've had bladder cancer. Then they don't currently have bladder cancer, according to the flexible cystoscopy, but the biomarker test is positive. And what we found, if we follow these patients up, if they're positive for molecular markers, they're 4 times more likely to subsequently have a recurrence. And the other thing that we've noticed is that in patients where we have a pair of samples, we have one before recurrence and one at recurrence, the levels of tumor DNA go up. So we're not as far advanced in the recurrence setting, but in theory, the ways we could take this test forward is it could be used to improve the scheduling of cystoscopy, to give more cystoscopies to the higher risk patients. It could potentially be used in the interval between cystoscopies to improve surveillance. It could be used with cystoscopy if patients are being discharged from their surveillance schedule. They both negative for our test and for cystoscopy, it might be safer to discharge them. Could we ultimately replace some cystoscopies, and could we do clever data analysis looking at the longitudinal biomarker trends in individual patients? So this is all future work. Where we are at the moment is that the test is now commercially available via Nonicus, and we are working to make this into a proper certified diagnostic test. Thank you very much, everyone, for your attention, for your hard work, everybody who's been involved in the project, and for your support. And with that, I'll pass you over to Duncan Shepherd, who is a professor of engineering, who is going to tell you about something completely different.
Right, good evening. Oops, come out with that. There we go. So I'm going to talk a little bit about biomedical engineering and what it can contribute towards in bladder cancer research. So biomedical engineering is the application of engineering to medicine. My background is as a mechanical engineer, so my research is about applying mechanical engineering techniques to medicine. And in terms of bladder cancer research, two of the areas that engineering can be used for is one is looking at the mechanical properties of bladder tissue, so understanding how stiff they are, how they deform. And the other area is in the design, manufacture and testing of medical devices. So I'm just going to go through a couple of areas. Firstly, the mechanical testing, and talking a little bit about devices. So in a lot of areas of mechanical engineering, we need to know the mechanical properties of materials. It could be a stainless steel, it could be a polymer. But you can also use these techniques to look at tissues. So on the left here, we've got a materials testing machine. So this is basically a machine that can either compress or apply tension to different materials. And we've used this technique of compressing materials to actually look at the mechanical properties of bladder tumour. So these are samples of bladder tumour here. And we basically put it into the materials testing machine and we compress it over a period of time. And from knowledge of the force that we've applied to the tissue and also how much it's deformed, we can get an understanding of its mechanical properties. So these are just some results. This is a graph of storage modules. It's effectively how stiff is the tumour against frequency that we test at. But it's more about the numbers that we get out of it here than any trends in the graph, because we can use these numbers, understanding the stiffness of the tissues. We can develop computer models that more accurately represent, for example, testing of devices. Or we can use the knowledge of the mechanical properties to develop phantoms. So these phantoms could be used for training surgeons, or it could be used for testing different surgical instruments. And then looking at some design work and testing work around the transurethral resection of bladder tumours. We've been working on a device that actually fits over the resectoscope to prevent particles of tumour from going off and reimplanting elsewhere in the bladder. So as engineers, we like designing things. So this is using a technique called computer-aided design, where we can use 3D CAD software to actually visualize designs. And to a certain extent, we can do some computational testing as well. So the idea behind this device is that it goes into the bladder and then it opens up to cover the tumour that you're operating on. And we've got these wires here that just open up and stretch this polymer leaf. So the engineering challenge here was obviously the size of the device to fit into the bladder, but also how you can open up the wires to actually create this umbrella type device. So this is a very, very early stage prototype. We like to go into the laboratory and just mess about with things and try different ideas out. So what we've ended up using here are these wires are manufactured from a material, it's a memory shape alloy called nitinol. And if you pass a current through it, you can effectively train these wires to go into different shapes. So you can see here we've got the device unopened. If you pass a current through the wires, they heat up and they start to open up into the device that we're looking for. We've then gone on to work with a company called Cambridge Mechatronics, who have a lot of experience of using these memory shape alloys. And we've started to develop a device that's obviously a bit more refined than the early stage prototypes. And you can see here, we've got the memory shape alloys embedded within a plastic. And as you pass a current through, it actually opens up. We then, in the laboratory, we can go through a series of testing, opening up the device, place it against a porcine bladder to look at the seal of the device, and so on. So hopefully that's just given you a bit of insight into what engineering can offer bladder cancer research. And I will now hand over to Roland.
Thank you.
So can you see my slides? Good. Okay. So welcome again, everyone. So I'm Roland. I'm the team lead for bioinformatics and genomics, and I want to talk a little bit about what I call the dark matter of transcriptomics. I will now tell you what I mean by this. So before I start with this, I just want to remind you what, for me as a bioinformatician, what are questions people ask me. And for this to understand is that the cancer is progressing. So we have early lesions, late lesions, and also we have different types of bladder cancer. One is a non-muscle invasive, and the other one's a muscle invasive one. And one big question is, looking on the molecular level, can I distinguish them with genomic and bioinformatics approaches? Can I perhaps find unusual molecules which tell me, for example, I have a grade 3 tumor? And of course, with the hope as we to find potential novel biomarkers and have to do less of these painful procedures. Problem is, as mentioned already, with bladder cancer, we don't know so much about all this as for other cancer types. So now let's go to the transcriptomics. I like to look at this. So we heard that cancer is always a disease of the genome, and the genome is something like a map or like you know, a plan, a traffic plan is put in here. But this is like a static plan. And what really happens in the cell is that genes are differently activated depending on the cell state, the tissue type, but also the disease state. And what I really want to look is into this. So how much traffic are these genes producing? And is that different in that lane or that lane? And that might be one cancer, that might be another one, and I start comparing. So this people do a lot, or bioinformaticians do a lot. It's called transcriptomic analysis or differential gene expression analysis. But what we want to do, we want to look if one of these cars is perhaps special, unusual, does something strange, right? So this is what I mean with the gray area of transcriptomics, or the dark matter. So what I'm looking at is a process called alternative splicing. So if a gene is activated, it produces something called the messenger RNA. The messenger RNA should be now quite well known to you as a type of molecule because it's in some of the vaccinations, right? And on the genome, the gene is organized like Lego bricks. So these Lego bricks can be sorted together in different ways in a process called splicing, and then produce from one gene a lot of different so-called isoforms. Yeah, this is important in higher organisms, important process to get diversity, what the cell can do. So of course, stuff can go wrong, and in cancer, everything goes wrong. So there is things what we call missplicing. And different Lego bricks can be used differently. They might get different boundaries and change the properties of proteins in the end. So this is what we dig in. So a special, really, really weird thing is, so normally splicing has a direction. So the blue brick always comes before the yellow one. The yellow one might or might not be used, and then there's the red one. So what happens sometimes is that we have a wrong way driver and things go backwards. And what happens is a process called backsplicing, and this produces something called a circular RNA. So for many years, people thought this is just transcriptional noise, but without going into detail, it turns out more and more that some of these circles, and there are many of them, many different ones, actually do something in the context of cancer. It might even be with important roles, like suppressing or acting as tumor suppressors or the opposite. So they are very stable molecules. So this is exciting because it might be good biomarkers. So they might go into the urine. And the question is, can we basically use them to detect disease or disease state? So, so this very big picture, this is an actual study we did. So, bioinformaticians always like to make something called a heat map. So this basically just shows the relation between different entities. And here, so you have circles which we found differentially active in different grades of bladder cancer, non-muscle invasive disease. And here on the top, you see that basically we can, depending on the activity, we see different groupings of cancer. And you see an interesting subgroup here of G3 cancers, which have a certain activity of these circles, which is exciting because it's something happening regularly. And so this means potentially we can exploit this further, right? So now if you give me a transcriptome sequencing, I can look at the circles. Nobody else does this in the UK, and I can tell you that it might fall in this group. So this here just tells you that certain cellular functionalities are enriched here. So this is mostly cell cycle and other things which are typical dysregulated things in cancer. But in this other group, actually, we don't know what exactly is going on, but the circles behave regularly. The nice thing is we could already see that some of them are actually shed into the urine by comparing it to other studies, which also looked at circles, but not in bladder cancer. So there might be an interesting way to go forward. So that is already everything I wanted to tell you to get like an idea of what really unusual things we can look at and do look at. So we work very closely with Doug, who and his team. This is a biomarker team, but because obviously there are some routes towards biomarkers, potential biomarkers. And these are the projects which are going on. We have a wonderful PhD project where we look at a certain complicated gene family, which has a lot of different splicing events and isoforms. And it does look that we can perhaps make an RNA-based assay. So it's a very promising candidates, but especially here is that we had to figure out how to best measure that in urine or in the cancer itself even. And so we came up with the idea, we don't look at one isoform, but how each isoform relates to each other. So this is, so Nada is a PhD student, which really did fantastic work here. And then Anshita is a postdoc in the team, very experienced bioinformatician. So she did all the circle work and is also now looking into other genes if their isoforms might be useful for us. So then there's Minghao, and with Minghao, we want also to investigate are all these events telling us something about important cancer biology? Like, for example, some cancers escapes the immune system, so might usage of isoforms or splicing play a role here. And we have a lot of students projects where we already test now and develop methods how to find the circles in urine best in our settings. And I want to emphasize before I hand over to the next speaker that the center is not only great in science, but also in education. So that's why I listed all the PhD students, and we have many great master and bachelor theses out of these projects. And so hopefully we are educating the bladder cancer researcher of the future. Okay, so now I want to hand over.
Roland, I'll just dive in there. So that's a really, really important point. Thank you very much for highlighting, is that yeah, it is important that the research centre trains the next generation of bladder cancer clinicians and scientists and clinician scientists. And also urology generally has quite a low profile in the undergraduate medical degree programme all across the UK and farther afield. And we're hoping that the research centre, the Bladder Cancer Research Centre, might be able to start to impact upon the undergraduate course as well. Thank you very much. And now over to Farhat, please. Yeah. Thank you, Roland.
Hello, everybody. Lovely to meet everybody. And again, I just will reiterate the messages from everybody to say thank you. Can you see my screen? Yeah. Is that the right format? Let me just put it into slideshow. Oh. Can you see that?
We're just waiting. You're in speaker view.
Okay, here we go. Let's put it into that one. Is that the one?
Yeah.
That one. Is that the one?
You're there. Perfect. Thanks, Farhat.
Right. Thank you very much, everybody. So I am the lead for novel therapeutics within the Bladder Cancer Centre. And I just want to really give you an update on where we're at. So Obviously, what we're interested in doing is developing novel therapeutics for bladder cancer. That's what we need. And what we would like to do, and I think Michael beautifully introduced this to us already, was to develop what we call a bladder cancer treatment matrix, which is where we have a panel of drugs where we're able to choose the best choice of drug for each patient based on the gene alterations within their tumours. And my name for it is, we call it the bladder cancer treatment matrix. If in simplicity, what it would look like is that Roland and Doug both have talked about the genetics. So your tumour, using the companion diagnostics in your urine and your samples, we would be able to work out what genetic subtype tumour that you have. And then we will be screening for drugs, and we identify panels of drugs. And critically, what we are doing in the lab is working out how these drugs work against the different subtypes. And in the ideal world, what would happen is that somebody would come in, the sample would be typed, and then we can look at our treatment matrix and say we have somebody who is an M4 type, and therefore drug number 1 and potentially 7 is likely to be useful for this particular patient. Now, we've already started this process off, we've already started screening, and what we already know is that some of the drugs that we have are very specific. They work on some subtypes, and some of them can be quite broad acting. And both of them have a place in the treatment portfolio for bladder cancer. So in terms of what we want to choose, we don't want to throw away the baby with the bathwater, as they say. We already know that we have some treatments like mitomycin C, for example. What we may want to do with mitomycin C, of course, is reduce the side effects associated with putting so much of it into the bladder. So can we reduce the dose? And can we enhance its activity by reducing the dose? The other area that we want to look at is that we want to look at the genetics. So Doug and Roland have both talked about the genetics. In every single cell in our body, bar the red blood cells, we have 2 meters of DNA in every single cell. And that is wrapped in, we have 50 trillion cells in our body, so we have 100 trillion meters of DNA in our bodies. That DNA, so the genetic code is, if you like, related over here, but the DNA that then gets folded up, wrapped up into these structures to form your chromosomes. And we can have changes and modifications over here that we call epigenetic changes that regulate gene expression. Now, what we know is that in bladder cancer, more than 70% of bladder cancers have mutations in at least one chromatin regulatory gene, i.e. genes that regulate how your DNA gets wrapped up and expressed. So working with the University of Oxford and the Structural Genomics Consortium, we have access to hundreds of molecules that are potential drugs, and some of them are existing drugs that can target some of these changes in bladder cancer. And in addition, we're looking and always horizon scanning to see what other molecules are being used and developed by other people to see if they have space within our model as well. So what we do is very simply is we take tumor cells. Now we have cell lines. These are cells that are growing in the lab and have been growing for quite a long time. But equally, we have now started growing patient-derived cells. So when somebody has a cystoscopy, or we can take some of those cells and we can start culturing them in the lab and really see how they respond to our drugs. Now we can grow these cells in what we call 2D culture, which is basically a flat plastic and the cells stick down. And it's a very powerful, useful tool. However, that's not what the tissue looks like in our bodies. And the other way that we like to grow our cells in what we call 3D cultures. 3D cultures are basically where the cells are able to form substructures, which are basically 3D. So these are 2 cell lines which have been grown in 3D culture. And this is actually an organoid grown from a patient sample. So we have established that method in the laboratory, and we can take these 2D and 3D cultures and we can put drug on them and we can see how they respond. Importantly, we need to think about dosing schedules. What we want to do is treat patients as early as possible. So in an ideal scenario, what we want to do is be able to treat patients with non-muscle invasive disease so that they don't progress to muscle invasive disease. Okay, and we know that we can give intravenous therapy, so we can inject the drug into your bladder. But the obvious point here is that the exposure time is only about an hour. You hold your bladder, hold your pee for about an hour, then you go away and you pee it out. And so the drug at high dose is only really around for about an hour. So we need drugs that can work if they're only hanging around for an hour. And likewise, so what we've done in our experiment is looked at what happens is when we put drug onto our cells and leave it on, versus if we put it on for an hour and take it away. Are the drugs still effective? And the short answer is that we have screen hundreds. And so if we look at there, this is along the bottom is basically drugs, just a selection of drugs. Each bar is a drug. Along here is survival. So 100% means that all of the cells are alive. Over here, nothing has died. And obviously down here, I mean, everything is dead. So when we put drug on and leave it on, we can see a lot of our drugs in this panel are demonstrating some degree of activity. Over here, on the other hand, when we use the 1 hour exposure treatment in 2D culture, we find that now many of these drugs aren't as efficient or effective, but actually some of them still are. And we start now developing candidate drugs. This doesn't rule these drugs out, because what this means is we need to think about formulations of drugs which would stay on the tumor cells within the bladder and actually have a longer exposure than just the 1 hour. So here are just what some of these images look like when we're analyzing them, so we can look over time. And a normal tumor organoid will grow over time and get bigger and bigger, but we can see with some of these drugs, BC1, BC2, they're starting to die and fall apart. Here we have mitomycin C, and when we combine them with mitomycin C, we can see that some of these are looking very unhappy and die quite quickly. So far, I can tell you that from our cell line work, we have got 10 interesting candidates, of which one of them is quite well developed. We are now validating those candidates against patient-derived 2D and 3D cultures to demonstrate how effective they are against the patient samples. We will be mapping that against the genome alterations that we are in these patient samples. And what we will be doing, and are starting to now do, is to populate this treatment matrix. At the same time, in the background, we're also trying to work out how do these drugs actually work, because by understanding their mechanism of action, we can optimize the treatments, and it also informs us about the biology of bladder cancer so that we can better develop treatments. So I'm going to stop there, and I want to say a lot of the work that I've shown you has been done by Kelly Ward, who's a urology trainee in our lab, who's actually a clinical academic, and hopefully will be joining us to do a PhD, and Yao, who is a postdoc in our lab. So I just want to stop there and say that in all that we do, we think about what it is that we're trying to achieve for the patient. That is fundamentally important to the message and the ethos of the centre. And we will continue to do our very best, and likewise we will continue to need your support. So I just want to say a huge thank you from all of us. And now I'm going to hand over, let me stop sharing, to Prashant, who is a consultant urologist and the lead for clinical development. Prashant.
Hello, hi, good evening. I hope you can all hear me, and can you see my slides? Great. Thank you, Farhad, for your introduction. Essentially, I'm a urologist. I would highlight particularly to Professor Adams, the only accredited urologist who is affiliated with a university appointment in the whole of Midlands. And I deal with all urology, let alone bladder cancers. I also happen to be the lead for urology, the clinical service for the whole of Birmingham, which is looking after approximately 2 million Brummies and doing all the service provision and arrangements throughout the UHB Trust. And it is an absolute privilege to work under the leadership of Rick and the vision he has and shares with all of us in bladder cancer. And I'm the theme lead for clinical research and epidemiology. So we've heard a lot today from instrumentation, from Duncan, and about a procedure called TURBT, which is essentially putting a camera in the bladder and then cutting it out using electric current. And this is how most of the bladder cancers are essentially removed and even staged. And as you can see in the picture here, the camera is inserted into the bladder. This is under general anesthesia. And then subsequently, it is cut away in small chunks and eventually all throughout deep enough so that the whole tumor is taken. And just to highlight, if you can see the slice of bread here in this picture, the bladder is even thinner than this. So in the picture, you might not appreciate how thin the bladder is. And we have to be really, really careful that we don't make any holes whilst doing this procedure. But we think that in case of bladder cancers, this whole pathway of putting a camera first, looking at the cancer, then the operation, which is TURBT, I've just explained, then leading up to staging of bladder cancer and then treating is a very primitive approach in dealing with bladder cancers, because in most modern cancer therapy, it's the other way around. You use imaging, okay, which is non-invasive. You take small little biopsies, stage the patient, and then treat the patient. So why can't these principles be applied in bladder cancers? And one can see over the decades in throughout Europe and even different countries that the survival from bladder cancer has essentially remained flat. There's another study as well, which looks at 30-year survival in bladder cancer, and not much change has happened. Healthcare has advanced so significantly in all other forms of cancers, but bladder cancer is one of the only cancers where the survival has remained flat. And do you know why? Because if we keep doing the same thing, we will get the same results. And I have a big issue with TURBT. Our group has a big issue with TURBT, and we are asking some fundamental questions through clinical trials about the value of doing a TURBT. If breast cancer specialists were urologists, then can you imagine what they would do with breast cancer? Stick some needles in, put a hot wire and core out the cancer, leaving the cancer behind at the edges. No one seems to do that. So why in urology we're doing this? And these are really fundamental questions, very difficult questions to answer. But luckily, we've got a group together. And the main purpose of the TURBT, what everyone says, is for diagnosis, for staging, for treatment, and for even palliating of the symptoms to a certain extent. Now, this is true in non-muscle invasive bladder cancer. These are the superficial type cancers, the low-grade ones, where you can achieve diagnosis, you can achieve staging, you can achieve treatment, as well as you can palliate the symptoms, mainly the blood in the urine, hematuria. Now, as I mentioned, even the technology is not yet changed, and I hope Duncan and his team can help improve the technology through his work in biomedical engineering. But in the meantime, the biggest harm we are doing to our patients with muscle invasive bladder cancers, the more advanced type of bladder cancers which are presenting. Luckily, it's not many, it's about 20 to 25%, but there are still very significant chunk of patients throughout the country, throughout the world with muscle invasive disease. So the TURBT is helpful in diagnosis, yes, certainly, because you get some tissue. Staging, maybe partially, because in muscle invasive, you can't keep digging in and making a hole, because if you do that, you will make a hole through this bladder and all the cancer is going to spill out from here. So it's dangerous. As far as treatment is concerned, you actually delay the treatment because there is another pathway, there's another procedure introduced inside so as to achieve a diagnosis. And finally, as far as palliation of symptoms is concerned, it is questionable. So on the basis of this premise, we set up the trial. And one thing I'd really like to emphasize here, that delay in diagnosis and delay in treatment can cause lives. Here is a very simple example. So a 2-week delay, and you can see the curve on the top and the curve at the bottom. They all separate out. And if you monitor these patients over years, there is a consistent and prone benefit of diagnosing these patients who present with the first symptoms as soon as possible. So delay should be avoided. We also think that the procedure can be harmful. As I said earlier, we have got complications from an operation, which everyone knows. The biggest risk here is making a hole in the bladder. 5% of the patients will make a proper hole, but in 50% of the patients, there is a small hole. And a problem with those things is when we are irrigating the fluid inside the bladder, it's under pressure, and the cancer cells can potentially spread to other structures, neighboring structures, or even in the bloodstream. And there are studies there to prove that circulating DNA, there's a sharp rise in it at the time of the operation. And we even postulate, along with other groups, that TURBT may actually be spreading cancer. So on that premise, we went to the NIHR and asked for some money. And we got a good chunk of money, and we got all the collaborators together, made a nationwide trial. A lot of work went in from the Bladder Path trial group, and we approached patients who had blood in the urine. We then asked them to participate in the study. They were randomized into two groups, which is TURBT versus the alternative route which we are proposing. And then in that alternative group, patients who were thought to have higher stage disease, i.e. muscle involvement, they then got an MRI scan. And on the basis of the scan alone, we decided whether they can go on to have their definitive treatments, which could be anything in the form of surgery or radiotherapy. But we eliminated the use of TURBT in that group of patients. Primary outcomes from that from the trial study is to see whether such a study is feasible, it is safe, how much time it will reduce to the primary treatment by eliminating the TURBT, and does it have an overall impact on failure-free survival. We have got some preliminary results which we published in European Urology, and the early impression what we get is the MRI scan does help in distinguishing between muscle-invasive disease and others. Our team has got confidence of actually giving treatment without having to resect the specimen through the operation called TURBT. We have stopped the trial recruitment in December, or just about a month ago, because we have achieved our cure of patients. We are in the process of finalizing all our results, and this year we'll be very busy with accumulating all the results and preparing a manuscript and spread this news to the rest of the world of the feasibility of this whole project of the Bladder Path trial. We've recruited, this was at the first impressions when we published, we included about 100 plus patients who were randomized, but now overall we have 200 patients. A lot of patients have gone in, and detailed analysis is yet to follow. We conclude that, as I said earlier, that TURBT is potentially dangerous, it's outdated, and we should be using MRI scans. And more importantly, we want to join the cancer treatments in the 21st century. This is one of the only procedures which has been there since more than half a century. And we are hell-bent in trying to change this. But when we do these things, we have to convince our community, the urology community throughout the world. We've had so many questions raised by top urologists all over the world about the whole clinical trial. Some of them have gone on to say that old-fashioned surgery is here to stay. And this is only the urologists who are doing that. So your contributions in helping us with these trials, more trials, asking really, really difficult questions, challenging questions, will help us address the diagnosis, the staging, and overall the outcomes from bladder cancer. So these are the important questions which we have to address. And in the words of Gandhi, this is what we face quite often. They first will ignore us, then they will laugh at us, and then they will fight us. But here we are in a team who are all very, very strongly united to give the right message to the rest of the world. Next year, hopefully, we'll be presenting the results of this trial in much more detail. But we've got some other exciting trials coming up as well, as what the other researchers have mentioned. We're currently going through a finalizing phase of one of the trials, which is collaborated with the rest of the researchers across in the country, as well as the industry, about injecting certain type of drugs directly into the bladder cancer. These are for advanced bladder cancer patients, and then treating them with radiotherapy. Thank you for listening. Over to Rick.
Prashant, thank you very much. Yeah, the Bladder Path trial that we've obviously, many of us have been very closely involved with for a long time. It is yeah, potentially paradigm shifting. And when you make such a big change in practice, and potentially one that may reduce the role of the operating urologist somewhat, you often face some challenges. And we just have to wait to see what the final evidence when we've analysed the full accrual of patients. But equally, you know, what we have is the opportunity to overlay some of the urine diagnostic work that we're doing into the patient pathway, some of the work that we're doing in molecular subtyping bladder cancers, such that that patient pathway can be very, very refined and even personalized to some degree. So thank you very much for that. Right, so questions. Would anyone like to put their hand up to ask a question, or would anyone like to, is there a question in the chat? So I can see a question in the chat that asks, that's to Prashant, says, really great trial. Did you do the MRIs after flexible cystoscopy? Were small, likely non-muscle invasive tumors excluded from needing MRI? So I can answer that. So it's a really good question. So the MRIs were done after flexible cystoscopy, and at flexible cystoscopy, in as many cases as possible, we took a biopsy via the flexible cystoscopy, and actually that's one of the key findings of the study, a sort of side finding of the study, that although it's often frowned upon to take biopsies at flexible cystoscopy, we found we can do it quite easily, and that the pathologists can still get a lot of information from the very small biopsy that you can get from a flexible cystoscopy. And then at the time of cystoscopy, urologists scored the tumors on a 5-point Likert scale as to the likelihood of them being muscle invasive. So actually, by looking at a tumor, urologists are very good at spotting those tumors that look invasive and those that look non-invasive. And score 1 were the patients who were definitely non-invasive. 5 were those that were definitely muscle invasive in the view of the urologist undertaking the flexible cystoscopy. And so it was the patients who were Likert score 3 to 5 who were eligible for randomization. So I hope that answers that question. Thomas Snell, I see you have a hand up.
Hi there. Yes.
Good evening.
Hi, good evening, Rick. Thank you for letting me speak, and thank you and to all the speakers this evening. It's been an incredible talk. It's been really insightful. So my question is, how can we on the call help take the research to the next step? What do the support, or sorry, what does the support and donations you receive enable you to do moving forward from here?
Thomas, that's a really helpful question. So thank you very much. So, you know, any amount of support is obviously very greatly appreciated, and the real advantage, I would say, of supporting a university research team, whether it be us here in the Bladder Cancer Research Centre or other teams in medical research across the medical school, is that those donations go directly into the laboratory. Is that you are supporting teams that are already up and running. They have staff in place who are doing all of this research day in, day out, and the money that you can donate will go directly into that, either laboratory-based research that you've heard a lot about, or the in silico research that involves a lot of analytical work and coding, the bioinformatics work that Roland leads on. You know, I think priorities for us is some work around how we realistically can implement both our diagnostic urine test within urology and also molecular subtyping. So using RNA to further classify tumors such that patients get the right treatment at the right time. And those are a big, again, another game changer, paradigm shifting work that has been essentially forgotten for 30 years. And so we have two generations of urologists to convince that these new ways are better. And so some pieces of research around convincing the field that these molecular approaches are beneficial, as they have been shown to be in very many other cancers, but convincing the field and patients that these are advantages is important. You know, I think there's a lot more to learn about the dark matter of genomics, Roland's work. There's a lot more to learn about cancer immunology. We already know that it's hopefully most of the general public now, after the last 2 years of COVID, appreciates to some extent the complexity of the immune system and the different strains of COVID and the different nature of the vaccines. You know, the immune system is incredibly complicated, and it has a big role to play in how our own body reacts to cancer cells and how our treatments affect cancer cells. And so we need to do a lot more to understand tumor immunology, and it's some work that we're doing already in partnership with Anna Wilkins, who I see from the Institute of Cancer Research, who also has her hand up. Anna, fire away.
Thanks so much, Rick, and thank you all for an absolutely fascinating hour. It's been brilliant to hear. I wonder, this is really kind of for Rick and Prashant, I think. You've spoken about the kind, successful approach of the flexible cystoscopy biopsy, but I wondered what you thought would be the best way ever to biopsy, in a way that you could perhaps do some kind of multi-region sampling, so you can potentially look at intratumoral heterogeneity. You know, with your clever surgical engineering hats on, what should we be trying to do as the best possible ever alternative to TURBT? Love to hear your views.
Prashant.
Very, very good question. I think you're just further giving us ideas on how we can do these things. So a very good point. The bladder cancer is heterogeneous. For the non-scientists in the group, it means that the cancers or the mutational signatures and things are very different in different parts of the bladder. So if you consider this room as the bladder, there might be different changes happening where the window is, there might be different changes happening where the door is, and so on and so forth. And how can we capture all that information in one go? It's a very, very good question. I think there is possibility. I think with Duncan's umbrella type of a cystoscope design, we can perhaps add pinches right at the tip of the Nitinol wires, which can probably biopsy the bladder at different parts. So we have to develop these technologies in stages, and so there is a possibility there. What Doug and his team has demonstrated, that we can now extract DNA from a urine sample, from a very small pellet involving only nanograms of DNA material, to have good quality readouts. So it's not the volume of the sample which is important, is how we collect it and from different areas. And we have demonstrated that it is feasible, and from the bladder path we'll probably get more information. I think what we want to head to eventually, using all these molecular strategies and mutational signatures from urine, is to have a smart toilet. I would like to live this through and see this invention come, hopefully through our center and through our collaborators, that one fine day, if you know and familiar with Birmingham, we'll go to Broad Street, install these smart toilets, pee into the machine, and it'll come out with a barcoded printout, which you can take it to your GP and have it analyzed there, whether you should be having a bladder cancer diagnosis or not. And that is the kind of vision we would like to take through.
Thank you. So I have been asked to maybe draw the session to a close. Alan, I know you've got a question. Because it's you, is it a quick one?
I think Prashant's just answered it. Thanks, Rick. As a patient, I've got a vested interest in all this stuff. It's been absolutely brilliant. I was wondering if one day we could look at population screening as opposed to just diagnosis when people have a symptom, because often bladder cancer, as you know, is asymptomatic. So
very quick answer to that is, so yes, some of our colleagues in South Yorkshire are already embarking upon that. We work with them very closely because clearly there's a role for our urine test within that, and it's just working out exactly how we achieve that. So those conversations are well underway. Alan, thank you very much.
Thank you.
Great, thank you everybody. And if we haven't managed to answer your questions, then I apologize, but feel free to email us the questions and we'll get back to you with an answer as soon as possible. Thank you all so much for joining us, and we hope to see you again. Well, it could be November. November would actually be 2 years. Maybe we shouldn't leave it as long next time, because this has been great fun. And as you've noticed, we do like to talk about the work that we're doing, and we have overrun, so I apologize for that. But great to see everybody. Thank you very much. Good evening.